It's like a dream come true.
I'm really happy to be here. I've learned about so many walls that can break.
You are dynamic, you have done earth-shattering stuff, and you're about to persuade us how to tear down the walls.
It can feel like the longest 3 minutes of your life, or the shortest.
Seeing everyone sitting there and looking at me was a whole new world.
To see them here on an international stage in front of so many prestigious judges and peers, it was incredible.
I hope that they maximize the opportunity and really smash the walls.
The room is like a powerhouse of thoughts.
People are really aiming for the moon with a lot of these exciting ideas.
So my goal of the project is to help people with Alzheimer's and to delay their pathology.
Please, raise your hand if you like magic.
Imagine a world without fossil fuels.
Really being able to select the best out of the best, that's probably the most challenging.
Well, a breakthrough winner should have had an original idea and a clever way of addressing it.
Impact on a global scale to have a sustainable solution.
For me, this peer group is inspiring me to make this world a better place.
The hope that clever young people with much talent can share with us gives us great optimism ultimately for the future.
Good afternoon, and thank you for joining us today at the home of Australia's science, the Shine Dome. And a big hello to all watching us online. We will have fun with falling walls. I'm Professor Hans Barffour.
And I am Dr. Emma Ann Carlson.
And we will be your MCs today. From us, more shortly. But first, I would like to welcome, to open the event, the President of the Australian Academy of Science, Professor Jagadish Shenupati. Please come to the stage, Jagadish, and tell us what this is all about.
Thank you, Hans and Emma. Good afternoon, everyone. I'd like to acknowledge and pay my respects to the traditional owners of the lands on which we meet, the Ngunnawal and Ngambri people. I also pay my respect to the traditional owners and the elders past and present of all the lands on which the Academy operates and its fellows and staff live and work. They are custodians of knowledge, critical to understanding our world and informing our decisions. Dr. Cathy Foley, Australia's Chief Scientist, Her Excellency Beata Jeske, Ambassador of the Federal Republic of Germany to Canberra, distinguished members of the jury, Falling Wall Slab presenters, fellows of the Academy, and event partners and colleagues, welcome. The very first Falling Wall Slab took place in Berlin on 7th of November 2011, marking the anniversary of the fall of the Berlin Wall. Since then, it has grown from strength to strength, with 78 labs taking place around the globe in 2024. And we are delighted to partner with the Falling Walls Foundation to deliver Falling Walls Lab Australia for the 9th time this year. Each year, on 7th November, 100 winners from the international labs gather in Berlin to pitch their breakthrough project to a global audience of industry leaders, decision makers, investors, and international media. We are lucky that Australia has once again been invited to send 3 representatives to the global finale and the top 3 presenters from today's event. Today's 16 presenters were selected in heats held in Adelaide, Brisbane, Melbourne, Perth, and Sydney. Falling Wall Slab in Adelaide, organized by the University of Adelaide and University of South Australia. Falling Wall Slab Brisbane, organized by Griffith University. Falling Wall Slab Melbourne, organized by Bio21 at the University of Melbourne. Falling Wall Slab Perth, organized by Curtin University. And Falling Wall Slab Sydney, organized by the Academy and Euraxis Worldwide, Australia and New Zealand. So the Academy would like to acknowledge these partners for their support and congratulate them on selecting such impressive finalists for today's event. So that means today's participants have already won at their state events. That means they're all winners already before winning and then going to Berlin. So the Academy is committed to modeling, promoting, and influencing best practice in diversity and inclusion in the STEM sector. We cannot maintain standards of excellence if we are not diverse. This means being proactive in removing barriers to participation. Doing so allows us to celebrate excellence from all the available talent across our country, unhindered by postcode, gender, culture, or other factors. For future state and territory Falling Wall Slab heats, we will incentivize participation of diverse candidates so everyone has a chance to participate and compete equitably. Finally, I would like to acknowledge and thank the following partners and sponsors for their generous support. Merck Group, the Embassy of the Federal Republic of Germany, Canberra, and Euraxis Worldwide, Australia and New Zealand. Your valued support brings this event to life and helps ensure the continuation of excellent and innovative scientific research and its application in Australia. I look forward to hearing from the 16 presenters and wish you all the very best of luck. And now I would like to invite Her Excellency Beata Jeske, Ambassador of the Federal Republic of Germany to Canberra, onto the stage to say a few words. Welcome, Ambassador Jeske.
Honourable President of the Australian Academy of Science, Professor Jagadish, distinguished jury members, dear Falling Walls, finalists and guests. I'm happy to see so many young researchers attending today's finale of Falling Walls Lab Australia. And this year, more finalists are taking part in this event as ever before. And thank you for being here and sharing your innovative ideas with all of us today. I'm proud that the German Embassy has been able to accompany this great event since 2016 and to observe its increasing popularity. And my gratitude goes to the Academy of Science for organizing today's event, Dr. Foley and the eminent members of the jury for giving their time. With the crumbling concrete walls, blocks of the Berlin Wall in mind, the Falling Walls Foundation was established 15 years ago with a mission to foster innovation that breaks down the barriers we face today. Whether these barriers are in science, society, or technology, the vision of Falling Walls is to create an environment where obstacles to progress are overcome and new possibilities are explored. This year marks a significant milestone as we commemorate the 35th anniversary of the fall of the Berlin Wall. And this momentous event had a profound impact not only on Germany, but also on the global landscape. The fall of the wall was a powerful symbol of freedom, unity, and the breaking down of barriers, both physical and ideological. It serves as a reminder that through determination and collective effort, even the most formidable walls can come down. Last week, I had the opportunity of traveling to Queensland on my official visit and visiting some of its universities. I learned much about their various research programs and their close collaborations with Germany. Of course, this was only a partial impression of the potential Australia has to offer, but these universities exemplified its excellent and world-leading research and university sector. And Australia's participation in Falling Walls Lab is characterized by remarkable success stories. In 2019, I hope I pronounce it right now, Pieri, back then at the University of Queensland, won first prize in the final in Berlin with his research on the reuse of waste glass. And last year, Dr. Emma N. Carlson, who is with us today, made remarkable achievements in both Australia and the international final, where she showcased her research on breast cancer and secured the 3rd position. I'm looking forward to witnessing the passion and potential of the bright young minds in this hall today. They are part of a next generation of leading scientists. Falling Walls Lab is a celebration of ideas that have the potential to change our world. This event brings together progressive minds from diverse fields, all united by a common goal, to break down the walls that hold us back and build bridges to a better future. In a world where, unfortunately, disinformation is on the rise, evidence-based research is indispensable. And Germany and Australia share a deep commitment to set evidence-based scientific research and innovation. Our two nations understand that the challenges of today, whether in healthcare, climate change, or artificial intelligence, require not only national but global options. The Falling Walls Lab exemplifies this belief, fostering an environment where the brightest minds can come together to solve the most pressing issues of our time. In conclusion, I would like to congratulate all the participants for being part of the Falling Walls Lab Australia 2024 and wish you the best of luck. May your ideas inspire and your passion ignite the next wave of breakthroughs. Let us continue to work together across borders and disciplines. Let's break down walls and build a brighter, more innovative future for all. Thank you very much.
Thank you, Your Excellency, for addressing us and for setting the scene.
Hello again, everyone. I'm so delighted to join Professor Hans Barffour as MC of Falling Walls Australia. As some of you may know, I was in the shoes of our 16 wonderful finalists this time last year, having recently won the Falling Walls Lab Brisbane a few weeks prior. So I have some sense of the excitement and nerves you may be feeling. It was such an honor to travel to Berlin with two amazing women and scientists, Dr. Alex Griffin and Himanshi Kyei, and I speak for all of us when I say that the support and mentorship of Hans, a longtime champion of the event, was invaluable to the experience. Falling Walls was a pivotal moment for my career and offered so many exciting opportunities, meeting new research collaborators, mentors, and friends. I met Nobel laureates, I spoke on the ABC Science Show, and I even got to see the famous capybaras at the Berlin Zoo. So presenters, I can't wait to hear about your groundbreaking work, and I wish you all the best. But first, a few housekeeping matters. Please turn your electronic devices onto silent, and we kindly ask that you don't take any calls during our amazing presentations today. And please also stay seated during the presentations as well.
Thank you, Emma. And it's great to see you back here. Now, for the full event program, you can actually scan the QR code. Yes, we have it up there on the screen now. We have actually a pretty tight schedule. Each presenter has just 3 minutes to present, and this will be followed by 2 minutes for responding to questions from the jury.
The jury will assess the presentations against the following criteria: breakthrough factor, relevance and impact, and finally structure and performance. Following the 16 presentations, there'll be an afternoon tea while the jury deliberates and decides on our top winners.
We are most grateful to the panel of eminent judges who will select the winners of today's presentation. And the judges are, and could you please raise your hand when I call you out, Dr. Cathy Foley, Australia's Chief Scientist who is today's jury's chair. Professor Jagadush and Beate Gierski, Her Excellency, who you heard already from on this stage. Michael Swanson, Director, Market Access and Government Affairs at Merck. Professor Peter Ashworth, Director of the Curtin Institute of Energy Transition. Professor Ross Hannon, Deputy Dean Research at the Australian National University College of Health and Medicine. And Dr. Li Gao, Deputy Chair of the Academy's Early and Mid-Career Research Forum and a research strategist at Southeast Water in Victoria.
And now we have the great pleasure of introducing today's presenters.
Dr. Stephanie Allison-Logan, co-founder and CEO of Sprout Materials.
Isaac Che Barffour of the University of Queensland's Institute for Molecular Bioscience.
Nicholas Berichev of the Australian National University's John Curtin School of Medical Research, Clear Vision Research Group.
Benjamin Kamer-Pesci of Murdoch University.
Dr. Natasha Del Avis of the University of Melbourne.
Dr. Jacob Martin of Curtin University.
Dr. Donovan Garcia-Cerron of La Trobe University.
Kira Picknell of the University of Technology, Sydney.
Saijon Ramesh of the Australian National University's Research School of Biology.
Dr. Harshal Patel of Flinders University.
Dr. Kelly Martinovich of Murdoch University.
Pratyush Ravichander of the University of Queensland.
Minjae Yang of RMIT Melbourne University.
Meher Shadwadia of the University of Queensland.
Dr. Jonathan Richetti of CSIRO, and finally,
Dr. Asara Sinaratne of Flinders University.
Now, this is a competition, and our main objective is to keep a level playing field. Our first, second, and third place winners will be decided by the jury and announced at the end of the event today. But we also have a People's Choice Award, and that means involvement of you. Those of you watching in person can vote for your favorite presentation on the slip of paper in front of you. I hope you have it, or you can get one. And pop it into the box when you exit the theater for the afternoon tea after the presentation. One vote only. Now, let's get started.
Please welcome our first presenter, Dr. Stephanie Allison-Logan, co-founder and CEO of Sprout Materials.
Think about the last thing that you purchased. Odds are it was either made of plastic or packaged in plastic. Over the last 70 years, plastic production has skyrocketed. In fact, over 50% of plastics were made in just the last 20 years alone. And this production is only going to continue increasing. In a lot of ways, it's not that surprising. Plastic is inexpensive, it's very durable, and can be used to make anything from furniture to foams to protect expensive and delicate medical devices and electronics, and even extend the shelf life of food through packaging. Unfortunately, our dependence on plastics is damaging our planet and our own health. 98% of plastics are made from fossil fuels, leading to nearly 2 billion tons of greenhouse gas emissions each year. Less than 10% of plastics are recycled, which means that they're building up in landfills, oceans, and even in our bodies as microplastics. At Sprout Materials, we are on a mission to develop bio-based plastics to replace conventional fossil fuel-based plastics. We have research developed at the ANU that we are going to use to change the world. We are using bio-derived materials to produce sustainable, home-compostable plastics. We start with bio-based materials that are available at large scale. things like plant oils and food additives. And we can also include biomass waste, such as spent coffee grounds, for increased sustainability. And from there, we can produce a wide range of materials. We can make plastics that are flexible or rigid, and we can even make foams that will return to their original shape after being compressed. These materials are home compostable in 4 to 6 months. They are made compatible with existing manufacturing methods, and because we are using large-scale commodity ingredients, we can actually achieve costs that are competitive with conventional plastics. So what's next? We are currently working towards our first commercial product, packaging foams, as a replacement for materials such as expanded polystyrene. We can use our foams to protect materials that are either expensive, electronics that are delicate, or even just materials that are heavy. We are estimating that our materials will actually lead to 30 to 45% less greenhouse gas emissions than the production of current plastics and their raw materials. These materials are completely renewable, and at end of life, they can be recycled and composted. And crucially, as I mentioned before, they are cost competitive, which is absolutely essential for ensuring that they can achieve widespread adoption. At Sprout Materials, we are working to break the wall of sustainable plastics, and we hope that someday you'll find our materials protecting your next purchase.
Thank you, Stephanie. Now let's go to questions. Oh, we're well ahead of time. Fantastic. So, may I ask the jury for the first question?
This is a really brilliant idea to solve a critical environmental issue. So my quick question is, when you mentioned that you have a lower greenhouse gas emission, let's see, 35 to 40%, but also you mentioned about the competitive cost. Can you give some quantitative measures? You are comparable to the current or traditional production cost, or you are a little bit higher. So what's actually the competitive cost mean? And also I would like to ask, what's the TRL technical or technology readiness level of your technology? Thank you.
Thank you. So in terms of costs, our goal is to produce the raw materials that will feed into plastics manufacturing, and we are aiming to make our methods compatible with existing manufacturing. So when we look at our carbon emissions and our costs, we're looking at kind of the cradle to gate. We estimate that our materials would cost us around $1.20 per kilogram when we're operating at large scale, whereas conventional plastics are currently around 90 cents to $1 per kilo for things like expanded polystyrene. However, we could also potentially address polyurethanes, which are often in the $2 to $3 per kilo for those raw materials. So while we are kind of high on pricing right now, based on estimates at purchasing at large scale, we think we can be about 30% more expensive than current polystyrene and less expensive than polyurethanes. It's also worth pointing out that with regulations coming in requiring certain percentages of recycled material, that those recycled materials are actually more expensive than virgin material, and that will also help our costs. And in terms of TLR, I'd say we're sitting at, I think, TLR 4 to 5. So we've made small prototypes, but we haven't yet done any pilot trials.
Good. Well, thank you, Stephanie, for the question session and for a very inspiring idea. Just as a housekeeper and timekeeper, we like to get 2 questions in 2 minutes. So sharp questions, sharp answers help. Now, please let me introduce, and we'll hear from Isaac Barffour of the University of Queensland's Institute of Molecular Biology. The stage is yours.
Have you ever had any severe pain that has taken you in and out of the doctor's office for the past 6 years but have not found the cause? That in those 6 years, you've been given a series of medication that you have holistically taken and have had a side effect but not cured to the pain? Well, this is the case of 1 in 7 of every woman in their reproductive years who are living with endometriosis, a disease that occurs when the lining of the uterus, called endometrium, develops outside the uterus. Endometriosis can manifest in one or a combination of the following effects: severe period pain, headache, fatigue, pain during and after intercourse, and infertility. Endometriosis is extremely heterogeneous but poorly understood. Because of this, patients have had to live with this terrible pain for 6 and sometimes 10 years before the disease is diagnosed. Currently, the only way of diagnosing endometriosis is through surgical laparoscopy, except that after 5 years of using surgery to treat it, 50% of patients develop endometriosis again, leaving patients to a lifelong loop of diagnosis, treatment, a brief period of relief, and disease relapse and overall deterioration in their quality of life. To break this wall, I'm using novel genome engineering technology and a small sample from tissues generously donated to the Endometriosis Research Queensland study to model endometriosis in the lab. And I'm using these models to simulate endometriosis treatment, helping us to assign the right treatment at the right dose for the right duration to the right patient. Data that we are gathering from the drug simulation process also allows us to identify gene markers and other molecular phenotypes. That is helping us to understand how endometriosis begins and naturally progresses. And we are also using this data to classify or develop effective endometriosis classification system that will help us to develop early and non-invasive endometriosis detection technologies. Endometriosis costs $9.7 billion in Australia alone for healthcare and loss of productivity every year. It derails the education hopes and career aspirations and affects the relationship dynamics and family outcome of patients. And I'm inviting you on a journey to the world where women suffering from endometriosis can have effective and personalized treatment. Thank you.
Thank you, Isaac. Now for questions from the jury.
Thank you for that really great talk, Isaac. It's a really important issue for many women. I'm just wondering if you could just tell us a little bit more about what was the breakthrough scientific aspect of the work that you presented today. What was the one thing that gave you an edge on being able to consider endometriosis that hasn't been done by others?
So one of the most important things that has delayed the progress of endometriosis is that the endometrial lining pretty much lasts about 24 to 35 days, and then it slots off and new one forms. So we have been able to go around that and grow endometriosis in our group for an extended period of time. And then the most important part of it is that I have been able to develop a genome engineering technology that has never been done anywhere else in the world. And this makes us possible to replicate the gene markers or gene mutations of every individual patient in our lab and try and see which treatment works for them, which has never been done anywhere else. Thank you.
Any other questions?
Really nice talk. I guess diagnosis is a real problem with endometriosis. A lot of it goes undiagnosed for many years. How will your work help with diagnosis?
Thank you. So the treatment, our group has been able to develop a drug screening pipeline. And so when I develop my models, we take it through the drug screening pipeline and see the current available treatment options and the one that works. And data that we are collecting from this study includes gene markers and other biomarkers like extracellular vesicles. That goes into the disease remodeling again. And because of that, we're able to determine that, okay, endometriosis, when it starts, it looks like this. When it's progressing, it looks like this. And we're able to trace all these gene markers along the line and use that one to develop non-invasive and early detection technologies.
Thank you, Isaac. Please welcome our third presenter, Nicholas Barashef of the Australian National University's John Curtin School of Medical Research, Clear Vision Research Group.
When I was 15 years old, I had a stroke and I lost part of my vision.
This is what I see. Our vision plays such an integral role in how we experience everything in our world, in every memory we make, and I'm willing to bet most of you can't imagine life without it. Now, the piece of tissue responsible for your vision, called your retina, actually damages as you age. And this damage happens at the most important part of your retina, which leads to a disease where your visual field will not look like what I see, but like this. This is age-related macular degeneration, or AMD. AMD is the leading cause of irreversible vision loss in Australia, and right now, there are no treatments that will stop this from happening to you. There is no stopping this disease. AMD is a big problem. My research is looking at how we can use something as easy as exercise to prevent AMD. Not only have I shown that physical exercise protects your retina against the same damage that causes AMD, but I have found a way to prescribe its molecular benefits without having to do any exercise at all. Just like we all here send important emails to each other every day, well, the cells in your body actually do the same thing. I've found that when you exercise, your muscles produce their own unique molecular emails, loaded with messages of good health. And these emails get delivered directly to your eyes. It's the messages inside these emails that's what's protecting your retina from damage. What is amazing is that we can actually hack into this emailing network, copy the messages inside these emails, and by doing so, effectively mimic the molecular response to exercise. I've shown that a single injection of these messages into an animal that has done no exercise at all shows the same level of protection as an animal that actually has exercised. By harnessing these messages, we can practically prescribe the neuroprotective effects of exercise to someone who is physically not able to. Using all the molecular material that our body already has, we possess the power to stop AMD and break down the walls of vision loss. Now I want you to take one more look at this image. This is what I have to live with every day, even when I look at this memory of me and my beautiful mum. This sucks. Living with this form of vision loss is what drives my passion for finding a cure for AMD, because if we don't do something now, this will be the future for 1 out of 7 of you. That's at least 12 people in this room. Using the molecular messages of exercise, my hope and my goal is to break down the walls of irreversible vision loss and help make this image clear for each and every one of you. Thank you.
Thank you, Nicholas. Now to some questions.
That's a great question. So a lot of research coming out in exercise research is looking at interval training, and it's the amount of exercise that actually affects the degree of protection that you see. And that's what we're actually seeing in our experiments we're doing, where we're exercising animals. We're actually noticing that it's the number of times that these animals are actually engaging in exercise that's actually contributing to the changes in these messages within these emails that we're seeing in the body. And that's exactly what correlates really well with what we're seeing in exercise research. Thank you for that question.
May I have a second question?
So these emails are produced by every cell in your body. I'm just calling them emails because they have another name. They're produced by every cell in your body. And I've actually shown that the purpose of these emails is just sending and receiving important molecular cargo in between cells. And I've shown that when you exercise, the molecular contents or messages within these emails actually changes in exercise as opposed to not exercising. And it's the messages in these emails that actually make their way all the way from your body to your eyes. And the overall goal with these messages are that you can administer them systemically and they will make their way to your eyes, as opposed to current treatments for AMD, which involve an intravitreal injection into the eye, which is invasive, costly, and overall just not ideal. So if we can overall harness these messages into a systemic pill or a systemic injection and prevent the onset of this devastating disease, that would be the dream. That's the goal.
Thank you. Thank you, Nicholas. Thank you, jury. Thank you, questionnaires. Good. Now, please welcome our next presenter, Benjamin Karmapesky of Murdoch University. Over to you.
Raise your hand if you love sushi. I do. My favorite part about sushi is the seaweed. I love seaweed because it's going to save the world. Pollution, hunger, and climate change. It can fight these three problems all at the same time. To the uninitiated, this might sound kind of crazy, so let me explain. Us Aussies, we love our Weet-Bix and milk, and we love a good steak. Because of this, we have a massive population of cattle in Australia. We've got about 30 million of them. We've got 30 million cows, more cows than we have humans. And these cows, they present a couple of problems that we have to deal with. Firstly, they produce a lot of nutrient-rich waste. And when they're concentrated in one area, this waste can have detrimental effects on our environment. They also produce a lot of methane. And methane is a really potent greenhouse gas, and this is a leading cause of global warming. So where does seaweed come in? Well, I'd like you all to think about sea lettuce. It's that leafy green stuff that grows down the beach. You see it all around Australia, and it feeds on the nutrients in the water. The more nutrients, the better. So what if we took the nutrient-rich wastewater produced from a dairy farm and we used it to grow seaweed? Well, this is exactly what I'm doing over at Murdoch University on the West Coast. When we grow seaweed in wastewater, we get two things. Firstly, the seaweed, it cleans up the wastewater. It pulls all the nutrients out, and it leaves us with clean water we can safely dump anywhere. And it gives us a lot of seaweed biomass because it grows very quickly. There's a couple cool things we can do with the biomass. It's been used as a food item all over the world for many, many years. It's good because it's high in protein and it has a good suite of micronutrients. We can also feed it back to the cows. And when we do this, not only do we create a circular economy of nutrients, but we can also reduce their methane emissions due to one of the carbohydrates that the seaweed produces. My continued research is about producing a high-quality seaweed product from seaweed grown in this way. When you change the conditions under which you grow seaweed, you change its chemical composition, which affects how good it is as a food, as an animal feed, or as a bioplastic or biofuel feedstock. Thank you.
Thank you, Ben. Jury, your questions, please.
Could you explain a little bit again what the novelty in your technological solution is compared with other solutions that are there with Seaweed, because Seaweed is already established in the discussion for solutions. So what's the speciality of your Seaweed?
Of course. So currently no one's looking at dairy farms in particular, and especially in the southwest where I come from, this is a really potent issue because of the pollution that we're getting in our waterways down there. So I guess the novelty for me is taking a couple different species of seaweed and trying to apply them to this area of wastewater treatment in particular.
On this, how much have you looked at the cost side of it? How much would it mean to implement it cost-wise?
Great question. So of course this is very important when it comes to practically applying this sort of technology. And I don't have an answer for you yet, but come back to me in maybe like 2 years and I'll let you know. I'm hoping that it'll be viable because of the products that we can produce from treating the wastewater and that the products we can generate with the seaweed.
Any other questions while we have time?
Yeah, so again, another good question. And again, this is something that I will be investigating. I don't have an answer for you just yet. But some of the pathogens and the microbes that you find in wastewater, they will only be able to survive in the fresh water. And when you bring them into the seawater culture of the seaweed, they won't survive.
Lovely. Cool. Thank you, Ben.
Thank you.
Please welcome our fifth presenter, Dr. Natasha de Alvis of the University of Melbourne.
Each year, there are around 200 million pregnancies worldwide. But sadly, we lose 800 pregnant women every single day. That's over 250,000 pregnant women every year. This is predominantly because most medications currently available on the market haven't been tested for their safety in pregnancy or approved for use in pregnancy. So the reason is that pregnant women have been systematically and repeatedly left out of clinical trials for critical life-saving therapies. Take COVID-19, for example. Pregnant people were excluded from the clinical trials for the vaccines and the antivirals, despite being one of the populations most vulnerable to severe disease. And the reason pregnant women are excluded from those clinical trials is for fear of harming the growing fetus. So we asked ourselves, what can we do about this? How can we get more therapies into clinical trials in pregnant people, more safe and effective therapies? And so what we've done is we've developed the world's first platform for testing the safety and efficacy of therapies for pregnancy. Over several years, we've developed sophisticated models of human pregnancy using precious human samples and animal models in which we can test the safety of new therapies and existing therapies. We've also developed specific models of pregnancy complications that are leading causes of stillbirth and premature birth. So in these models, not only can we test whether the therapies are safe, but we can also identify the most effective therapies at treating those diseases and take those to clinical trials. We're going to be working with industry to provide this as a service, so industry can bring their newly developed therapies into our models to rapidly test their safety for use in pregnancy, allowing us to make more therapies available to pregnant people quicker, and hopefully protect them sooner in the event of another pandemic. Our platform is also bringing together multidisciplinary expertise, from other researchers to bioengineers to industry to clinicians, to design new therapies specifically for use in pregnancy. For example, we'll be using new nanotechnology approaches to deliver medications to specific organs in the maternal system while keeping the growing baby safe. And this is a really exceptional step forward in our field where there's been a real lack of new therapeutic development. So what we're going to be doing is really de-risking clinical trials, choosing the best and the safest therapies to then progress, hopefully improving equity of access to therapies during pregnancy, and most importantly, getting more mothers and their babies safely home. Thank you.
Thank you, Natasha. And may I ask for questions?
Thank you, Natasha. And how accessible this technology is for the developing countries? You think if you once you develop this one?
Yes, so we're hoping that our platform can test all sorts of new therapies. We can identify their safety here and then publish that data to be used in other countries to change their kind of categorization of safety in pregnancy. In developing nations, what we're finding is really important is that there's low resources to treat pregnancy complications. So if we could provide more medications to say, okay, here you take this medication, go home for a week and then come back to us, it would really help in the sense that they don't have the resources to keep people and give them beds in the clinical settings.
Second question.
Nice talk. You're going to have to get FDA approval or TGA approval. How difficult do you think, what will be the key step to being able to actually go into humans from, avatars and models? The only area I know they're doing that is probably in childhood brain cancer, where they're going from 3D models straight into children.
Yep, so if you read around how we currently categorize therapies for safety in pregnancy, I think you'd be scared. Because the current categorization basically is based on what data we currently have of people serendipitously using these therapies in pregnancy. So they're categorized category A if there's lots of evidence of their safety, B and C if there's not much data to show their safety, and thus should only be used when the benefit outweighs the risk. And unfortunately, most therapies are in that B and C category. So we don't really have a way of currently getting drugs approved for use in pregnancy and then put into clinical trials and then put it on the population.
Good. Thank you, Natasha. So please let me ask Dr. Jacob Martin to the screen. Jacob is from Curtin University. All to you.
So I'm going to take a guess that, here we go, it's on. I'm going to take a guess that everyone has one of these on their person right now, a lithium-ion battery. These are going to be critical for us reducing our carbon emissions. They're going to allow us to store renewable energy on the grid. They're going to allow us to drive around in electric cars. But concerns have been raised about the safety of lithium-ion batteries. And I'm sure everyone's seen one of those viral videos of the whole, battery exploding violently. But here's the thing. I'm sure quite a few of you will go to sleep tonight with this right next to your head. So, how did we tame such a volatile element? The answer is on the screen. It's graphite. I've got a nice chunk here of graphite. Graphite is actually key for safely storing lithium. And this was actually what Akira Yoshino won the Nobel Prize for. He found that the layered structure of graphite allows the lithium to be safely stored when you charge your battery. The problem is that it's harder and harder to get this out of the ground and process it. We have to use some seriously toxic acids to literally dissolve the glass that is around this graphite. And so one option is to synthesize it. But there are huge issues with synthesizing it. At the moment, there's actually only a few oil wells around the world that produce the oil that can turn into graphite. You have to go up to 3,000 degrees. That's half the surface temperature of the sun. It takes 3 days to do this and an enormous amount of energy, usually derived from coal. So we wanted to ask the research question, can we make graphite green? We're physicists, so we turned to the coolest bit of kit we could find, and that is an electron microscope. And we wanted to figure out what's happening at the atomic scale. And you can actually see their atoms, and that defect there, we could actually target that and remove it, allowing us to make graphite in 10 seconds. All right? That's an order of magnitude quicker. That means we can use renewable energy. We can also use biomass, and we can take the defects out of biomass and create green graphite that way too. So we're currently commercializing this technology, and we're really hoping, my dream at least, is that we'll have factories right next to solar farms so that we'll be able to break the wall of truly green graphite. Thanks for listening.
Thank you, Jacob. Now for questions from the jury.
I love the idea of growing our own graphite. I'm just curious to know what is needed to be able to do it. You said you don't have to have the high temperatures and you don't need to use this particular special oils. You can use biomass or something like that. But what is it a low temperature method? Is it a chemical method? Didn't quite understand that.
Yes, so it's actually a chemical method. So we found a catalytic mix that could actually turn biomass into graphite. And it's actually the same temperature, 3,000 degrees, but we can do that very, very quickly. And so that's really important. And it turns out graphite's one of these materials that you can heat up the fastest of any material. So you can heat it up to 3,000 degrees in 1 second and it won't shatter, which is kind of an amazing property of graphite.
And what would you use to get that high temperature quickly?
So there's actually a number of different technologies that we're exploring. Induction is one of those approaches, but graphite you can just pass resistively an enormous amount of current through it as well. So we might just be able to use quite traditional heating methods as well. Good question. So there's a whole bunch of ways that you can degrade the anode. Actually, natural graphite has got a few issues with this. It can actually fragment. Synthetic graphite actually holds together a lot better, and it's actually those screws. It has super screws that hold the whole structure together. So it's actually not so much a problem with synthetic graphite. One of the big issues is making sure that the entire surface has a good reactivity so that it doesn't sort of keep eating the lithium up. That's the biggest issue. And also, it doesn't react with the electrolyte to produce the gases that lead to the swelling of the battery. So, but there's some interesting chemistry involved, and I'm happy to chat afterwards if you want to know more.
Thank you, Jacob. Please welcome our next presenter, Dr. Donovan Garcia-Serran of La Trobe University.
"50 farmers die each day due to pesticide poisonings. That's 20,000 deaths per year. Combined, there's 1 million cases of pesticide poisonings, and 99% of these occur in developing nations. We often don't talk about it, but we have a huge problem with pesticides. Realistically, we cannot phase them out today. To feed 8 billion people, there is an unprecedented demand for pesticides. But when these are used inappropriately, they lead to health issues and ecosystem damage. Australia and Australian research can do something about it. So the question is, how do we create better pesticides? Here's an idea. RNAi stands for RNA interference, and this is a natural process that living organisms use to regulate their own development. For example, imagine that you are at a gathering. There is food available. The spring rolls become more popular than the rice rolls, so the kitchen gets a signal. No more rice rolls, we need spring rolls. RNAi does exactly that for your body. Using an appropriate signal, it will change the production of your genes. And these genes are small instruction manuals used to build anything in your body. Now this is where it gets interesting. Scientists worked out what this signal was. It is a molecule called double-stranded RNA, and they could use it to essentially switch off genes in one particular pathogen at one time. This means that the instruction manual is no longer available, and the pest does not know how to cause disease anymore. How do we actually do this in the lab? We create an RNAi signal, and we spray it onto a plant. When the pest finds the spray, this will trigger the destruction of all the instruction manuals, and this means that the plant is protected. This technology is already being commercialized, but we need to expand it to protect more crops. It has major advantages, such as not harming beneficial organisms, not being toxic to humans, and being versatile. If we want to change it to other pests, we can change the sequences if we know which gene we want to target. My team and I are using it to protect grapes, strawberries, chickpeas, and lentils. We envision Australia as a powerhouse in the development of RNAi technologies to help agriculture. This is a new era in crop protection. Let's break the walls of food insecurity. Thank you.
Thank you, Donovan. Let's hear from the jury.
Thank you. Just two first quick question. Are you actually spraying the double-stranded RNA onto the crops?
That is right, yes.
So I guess my question is, RNA being quite labile, how practical will that be for remote areas? You know, chemicals are very stable. How will you get that to remote areas in Australia or Africa or places you might need it?
Yes, you're right. double-stranded RNA is more stable than other forms of RNA. And for the purposes of this talk, I didn't mention, but we encapsulate or layer RNA with nano layers called layer double hydroxide. So we essentially create these sandwiches of RNA, and when they are sprayed, they're protected from UV rays, from wind, from water, and they have a slow release, which means that the crop gets RNA released over time, and it has a duration on the plant of approximately 30 days.
Second question, please.
Right now, today, it's not comparable because of the scale at which commercial fungicides are produced, but with emerging technologies such as solar energy and others, the cost has dropped dramatically over the past few years, and it is expected with more adoption, it will come down to even $7 per hectare sprayed.
And final?
Double-stranded RNA, or is it, do you have to design it for the particular crop or the particular disease you're trying to control?
We can make different stacks, selecting different genes with the intention of targeting a vast array of pathogens with the one construct, but it will be on a case-by-case basis.
Good. Thank you, Donovan. Now, please welcome Kyei Pignell of the University of Technology in Sydney.
All right, I want to start with a question. What do beer and cows have in common? Now, I bet all of you have thought to the weekend barbecue, and that's certainly true. They do make a good pair. But I'm here to talk to you about how we can use waste to connect the two to help sustainably feed 9 billion people by 2050. So as the old saying goes, one man's trash really can be another man's treasure. But first, let's start with the fun stuff and chat about beer. Well, more specifically, how beer's made. You take some yeast, add some grain, the magic of fermentation, and essentially, you're done. But what do we get from this process? We get, one, a lovely chilled beer, but two major forms of waste in the form of brewer spent grain and carbon dioxide. It's pretty clear in Australia, we love a beer. In 2022 alone, we produced about 17 million hectolitres. So if we tally up Australia's bar tab, that's around 350,000 tonnes of grain and around 10,000 tonnes of carbon dioxide. Now, I'd place a bet everyone in this room's probably heard about carbon dioxide and how it's warming our planet. Do you know who else is racking up a pretty big bar tab in terms of greenhouse gas emissions? Our cattle and sheep. These guys are getting a pretty bad rap because they naturally produce methane, which is 25 times more potent than carbon dioxide. But here is the connection. By incorporating microalgae into the beer-making process, we can capture this carbon dioxide to produce nutrient-rich algae biomass for use in feed. So we're utilising this waste and throwing it instead of throwing it out as trash. So currently, right now, we have 2 bioreactors on the brewery floor that's capturing this carbon at a rate of 1.8 kilos of carbon for every kilo of algae biomass. This biomass is rich in nutrients such as protein and lipids, and we can combine it with the brewer spent grain to create a nutrient-rich feed for Australian farmers. Studies have shown that the inclusion of algae in feed can actually reduce the production of methane. So we set out to test different species' effect on methane production, and we're able to see that there was a clear species and dose effect, and we're able to reduce methane in the lab by an 8% reduction. From here, we formulated pellets to then use in a lamb performance trial to see how the lambs performed and how their health was affected by the inclusion of the grain and the algae. And our lambs thrived. They maintained intake, daily growth rates, and meat quality. So by using Australia's abundant waste streams that is created by breweries, one man's trash, we can create it into a high-value feed for Australian farmers to help them reduce their methane emissions, another man's treasure. Thank you.
Thank you, Kira. Now let's see what questions the jury have.
Microphone.
Microphone. How efficient is the process that you're suggesting when you're thinking about reducing emissions altogether?
Yeah, so the total, if we look at the total system, that's what we're currently optimising now. So we're currently designing a techno-economical analysis of seeing how we can look at those points where we're being obstructed. The main thing is cultivation, so the growth of how much algae we need. But the key thing is that we only have to use a small amount of the algae in the feed. So that's really helping us in being able to grow it at a capacity that we can then use it in the feed. Another thing for the efficiency is optimising the supply chain. So the great thing is that we have a lot of breweries around Australia, and we're looking at how can we optimise those supply chains locally for that brewery to supply to local farms, have that algae and that potential to reduce methane supplied in that almost like a little network locally.
And I guess being a bit more provocative, is there a reason, have you thought about actually just getting rid of the meat altogether and what that would look like?
I think that's a really cool part of Australia. We produce meat. A lot of our livelihoods rely on meat, and it's a really key protein in many diets. As well as internationally, it's quite an accessible protein. So I don't think the solution is getting rid of it. Eating it in a sustainable manner, yes, we can reduce our intake, but we need to find a way to support farmers, support those livelihoods in Australia and outside of Australia, because we'd lose a lot of jobs if we got rid of meat.
Any further questions?
Nobody else has one. Yeah. I want to know, do the lambs like it? You know, do they prefer it to grass or to other feedstocks?
Yeah, so the design was set up that we had a standard pellet with no algae, and then we put 2 different types of fresh and marine and see if they liked it. And so when I say they didn't reduce intake, that means they ate it at the same rate as what they ate the control. So if they didn't like it, we would see a reduction there.
Thank you, Kira.
Thank you.
Phew, we're halfway through and the presentation's been amazing. Please welcome our 9th presenter, Saisham Ramesh of the Australian National University's Research School of Biology.
We all love junk foods, don't we? Spicy McChicken, double cheeseburgers, filled with cholesterol. Do you know what loves cholesterol more than us? Parasites. Parasites that causes infectious diseases like Plasmodium, which causes malaria, or Giardia, which causes giardiasis. These parasites need cholesterol to make their own membranes, but they cannot make cholesterol and steal it from us, humans. Just these 2 parasites affected more than 500 million people in 2022, and the parasitic infections are soaring high due to drug resistance. I've used these parasites' love for cholesterol as a strategy to dodge these bullets. But first, let me tell you a short story. The Greeks were fighting a war against the Trojans and were about to lose the war. Then, Greeks hid inside a hollow wooden horse, tricked the Trojans, attacked them at night, and won the war. But with a lot of casualties. Had they used the Trojan elephant or a Trojan hippopotamus, they could have hidden more soldiers inside and reduced their casualties. Inspired by the Greek strategy, we tricked the 2 parasites I mentioned by conjugating, or let's just say hiding, anti-parasitic drugs within cholesterol. The parasites, greedy for cholesterol, took the drugs in along with it and were killed more efficiently than when presented with just the drugs. Even the drug-resistant parasites were tricked by this approach. I then wanted a better strategy, something that is even better for a wider range of parasites. So I kept the hidden soldiers constant and replaced the horse with 5 other animals. More scientifically, I kept the anti-parasitic drug constant and replaced the cholesterol with 5 other sterols, which are greasy substances closely related to cholesterol. Interestingly, one particular sterol with the hidden drug killed the parasites even more efficiently than cholesterol with the same hidden drug. While the parasites were naive and took the assorted Trojan animals in and got killed, human cells were smart enough to leave them outside, meaning our strategy is on target and does not affect the human cells. So far, we have concentrated on the human parasites. Next, I'll test this strategy on 4 to 5 veterinary parasites that causes over $10 billion in losses to the livestock industry worldwide every year. Redefining the Trojan horse strategy, I hope and aim to break down the walls of an array of anti-parasitic drug resistance.
Thank you, Sai. Jury members, your questions, please.
Oh, that was great. I'd like to know about how you're going to deliver this, because a lot of the people that are infected by parasites have very low incomes. And what sort of strategy for delivery? Will they have, 4 or 5 days that you have to take the drug? How much is it going to cost again?
So we're still working on the lab setting. So the parasites that grow, for example, the Plasmodium parasites that we grow for has a 2-day life cycle, so 48-hour life cycle. So, and the mode of action for certain drugs are faster. For example, artemisinin is only 1 to 1.5. This drug is the front line for malaria. But some other drugs have a longer duration. But with these sterols, the main advantage is the mode of delivery is quicker. So one advantage, it's quicker. And because it's quicker, the amount of drug that gets in is also more. So the efficiency gets higher.
And did you say I'm not sure if you said it, did you say the drug and the sterol are conjugated? Or
yes, they're conjugated, yeah.
Thank you.
May I have a second question?
For me, a lot of the problem with resistance is actually we don't have enough drugs. So you're planning to deliver them in a cool way, but it's still the same problem, is there not, in terms of the resistance, evolutionary resistance to the drugs?
Based on our results, we tested this strategy against artemisinin-resistant parasites. We found that it was 3 to 4 times more efficient, even for the drug-resistant parasites. Because we are using this sterol uptake mechanism, which the parasites cannot live without, this reduces the chances of these parasites emerging drug resistance towards it. And even the current drugs that are already in shows a higher efficiency when conjugated with these drugs.
Thank you, Sai. Thank you. And now, let's welcome Dr. Harshal Patel of Flinders University for the presentation. Green, push again. Yes. But then you speak into that. Or you use yours.
Hello.
Either way.
Polymers are used to build our homes, transport our goods, and electrify our lives. These fibers, rubbers, plastics, and foams are essential for modern life. Unfortunately, polymers are not made sustainably. Currently, we rely on non-renewable feedstocks to make polymers. After polymers are used, they are typically discarded in landfill or incinerated. This linear consumption model, make, use, discard, is wasteful and harms the planet. Incredibly, humans have made billions of tons of polymer waste. The situation is so dire that the UN is negotiating a treaty to combat this waste. A new strategy is needed to make polymers sustainable. In my research, I aim to break the wall of polymer waste. If polymers were converted back into their building blocks, this would drastically reduce polymer waste generation. In principle, we could make and unmake our polymers infinitely, but this is challenging with current polymers. The chemical bonds that join monomer building blocks to form polymers are very strong and hard to break. This property makes recycling current polymers extremely challenging. I have invented a new platform for polymers that contain new chemical linkages between monomers. Our polymers perform similarly to conventional polymers. The key difference is that these new linkages can be broken apart on demand when recycling is required. In this way, polymers can be made, used, and then unmade into their original building blocks. In my invention, sulfur-sulfur bonds link monomers together. The key linkage contains 3 sulfur atoms, a trisulfide. In collaboration with a team at Flinders University, we have discovered a new and highly efficient reaction to make these polymers and unmake these polymers. In this example, this new reaction was used to make a new analog of high-density polyethylene, a plastic used in many industrial applications. Unlike polyethylene, our polymer can be triggered to depolymerize upon the addition of a unique solvent. And so, a hard plastic can be converted back to its original building block in seconds. No waste is generated in this process, and all materials can be used and reused. Our aim is to apply the same chemistry to other major classes of polymers, focusing on those that are currently not easy to recycle. This includes polyolefin plastics, polyurethane foams, and cross-linked rubbers. The goal is to create a general platform for making, using, and unmaking the polymers that are essential to our daily lives. Thank you, and I'm happy to take your questions.
Thank you, Harshal. Jury, your questions now?
Hi, Harsha. This is a really great concept to produce a general platform. But my question is, you have now successful one, how feasible you can actually create a general, let's say, platform to produce different types of the polymers?
Sure. So this is very early days of exploring this chemistry. So we're still in the lab on small scale. So our aim moving forward is to apply the same chemistry to new classes of polymers, continue increasing the scale, and then to make these things viable in the future, have very targeted applications where the stewardship of these materials is well managed. For example, having a car where all the materials inside the car can be recycled.
And following on, I just want to ask, maybe clarify, you said, okay, you created a polymer and then they can reuse, but during your production of your polymers, how green of the process? You mentioned you don't produce any waste from the manufacturing process?
Slide. So the same component that we remove to drive the polymerization forward is the same component we add back for the depolymerization. So in that way, we keep using that same chemical to both create the polymer and unmake the polymer.
Real world setting, this new polymer, is any potential breaking down?
Sure, that's a good question. So far, with the materials we have made, they've just been sitting open in the lab, and we haven't had any issues with depolymerizing them. Of course, testing long-term world applications is going to require more testing.
Thank you, Harshal. Please welcome our 11th presenter, Dr. Kelly Martinovic of the Murdoch University.
Now, we all know about the dangers of high cholesterol and how a healthy diet and lifestyle can reduce these risks. But for every 1 in 250 people born with a genetic condition called familial hypercholesteremia or inherited high cholesterol, no amount of diet and exercise alone will reduce the levels of bad cholesterol in their blood. They are born with a faulty receptor on their liver cells that prevents them from being able to pull cholesterol from the blood and into the cell where it can be used. Instead, the cholesterol roams through the blood vessels where it accumulates on artery walls, forming hard plaques that lead to cardiovascular disease, heart attacks, and even death. Current treatment requires people to take a statin tablet every day for the rest of their lives. What we're seeing in clinic now, though, is people with particular genetic mistakes are not getting enough benefit from statins alone. They need new and innovative drugs that directly target and correct the faulty liver receptor so it can process cholesterol. And I've developed a drug that can do just that. My drug is an antisense oligonucleotide that corrects cholesterol processing by inducing exon skipping. So what is exon skipping? Imagine this stepping stone bridge represents your DNA. Each stone is a part called an exon that contains the code needed to make all the proteins in your body. When viewed together, they form a functional bridge or protein. For people with familial hypercholesteremia, it's like one of their stones is broken or mutated, and no functional bridge or protein is made. My drug tells the body to skip over that broken exon so that a functional protein can be made. Using patients' own cells, we have shown that my drug corrects the liver receptor's function. This is represented here by the increasing green signal in these images, which is cells taking up cholesterol. These liver receptors now work as well as those from an unaffected individual, as you can see in this graph. My research is providing hope to the millions of people globally who live with the constant threat of heart attacks due to familial hypercholesteremia. By harnessing the power of exon skipping, we can transform lives by reducing the need for daily medications to just a couple of injections per year. This breakthrough can redefine treatment standards by providing more effective and sustainable drugs for those who need it most. Thank you.
Thank you, Kelly. Now, 2 questions, please.
Thank you, Kelly. And so this need to be approved by the FDA, and then what stage this is in at the moment?
At the moment, we've got really solid proof of concept data, which I've just showed you. We're doing a few more in vivo studies to understand the dosing exactly, and then we'll go to formal preclinical toxicology studies and then a phase 1. The antisense oligonucleotides as a technology have had 5 drugs FDA approved for various conditions. The lab head that I'm in is responsible for 3 of those for Duchenne muscular dystrophy.
Okay, thank you.
Tested the level of cholesterol after you've administered this to a patient and figuring out, if it's 4.5 millimoles or where it sort of sits at the end?
Yeah. At the moment, we're still in the lab, just working out the dosing, and we're trying to also conjugate it to a GalNAc molecule, which tells our drug just directly to go to the liver, which means you can use 10-fold less. We haven't given anything to a patient yet. That will probably be in a few years' time once we've got some more data here.
Problems with all these is about delivery, and taking statins as a pill in the morning. How are you going to deliver this? Do you have to inject it or what?
At the moment, we think that our drug will be a twice-yearly injection, and this will be administered by the doctor, and this allows a pivotal point where the doctor can check in with the patient as well, check their cholesterol levels and make sure they're still getting enough benefit from our drug. The benefit of these twice-yearly injections is, like I said before, we can conjugate it to a molecule that takes it directly to the liver, so we don't need to give as much and we shouldn't need to give it as often. So that will also bring the cost down as well. Thank you.
Thank you, Kelly. And up next is Katyush Ravichander of the University of Queensland. It's all yours.
Imagine a world where the ability to feed a growing population is compromised. This is not a dystopian future, but a sad reality. By 2050, our planet is projected to house nearly 10 billion people, demanding a staggering 75% increase in food production. However, as we strive to meet this need, fungal diseases pose a significant threat to our food security. Fusarium head blight is one such example that threatens cereal crops across the world. This disease is caused by a pathogen known as Fusarium graminearum, and it can slash yields by up to 50% and contaminate the grains with toxins that are harmful to both humans and livestock. And to make matters worse, climate change is only amplifying the severity of this disease. While chemical fungicides are used, yes, they may be effective, but they are not a sustainable solution. This is because the overuse can result in the pathogen in developing resistance, but it can also cause harm to the ecosystem. This crisis calls for an innovation, a sustainable solution to protect our crops, food supply, and ultimately our future. Enter RNA interference, or RNAi. This is a biological process that occurs naturally in most living organisms. It functions like a pair of molecular scissors, resulting in gene silencing. My lab and I are working on one of its applications, which is known as the spray-induced gene silencing mechanism. We have developed a process to produce specialized RNA molecules in the form of a double-stranded RNA spray, which can target the essential genes of a pathogen. These genes are crucial for the pathogen's functionality, just like the foundation of a building. And so by silencing them, we can kill the pathogen and protect the crops. We have developed a way to enhance the efficacy and the durability of these molecules by combining biotechnology and nanotechnology to produce a super spray. We call this discovery as bioclay. Bioclay involves the use of biodegradable clay-like nanoparticles that encapsulates these RNA molecules. And by doing so, we can prolong plant protection for up to 28 days. I have designed and developed 2 RNA targets that have successfully been advanced in glasshouse trials. These targets have shown to significantly reduce disease incidence against Fusarium head blight in wheat and barley as a curative spray. But I am only one player in a team sport, which is my group. And together we are working on the same goal, that is to break down the walls of RNA-based fungicides to tackle some of the world's most significant fungal diseases. This is so that we can create a sustainable future and to ensure food security for generations to come.
Thank you, Pratyush. Jury, your questions, please.
Thanks for that. It's terrific. The problem here, I guess, is the cost. It's quite expensive to make, double-stranded RNA and all this sort of stuff. And we're talking about an industry where cost is really, really important. So do you have any idea of the cost effectiveness?
Yeah, sorry. Well, it's actually, I was reading a paper, and in 2021, the cost for producing double-stranded RNA has come down to $1 per gram. However, I think the solution to this is I know it is still expensive to produce double-stranded RNA, especially for developing countries, but to combine chemicals as well as double-stranded RNA, this reduces the sustainability issue. This helps us with the sustainability issue. So that's in the near future, I think it's best for us to combine chemical fungicides as well as double-stranded RNA.
About what the difference is from the non-clay and the clay difference. I didn't quite get what the advantage was and how you realise that.
So I know Donovan had already mentioned about this. Double-stranded RNA is stable, but the use of nanoparticles is to help increase the stability in the environment, in a natural environment. Yes, dsRNA maybe in this setting can last for 2 weeks. However, when you apply it in soil, it probably gets degraded because of the enzymes that may be present. These nanoparticles help to encapsulate that, and by doing so, it helps survive in the soil and makes it more stable in the soil for longer. But not only that, it also helps in increasing the uptake within the plants as well.
Because it breaks down or something?
No, it's all biodegradable. That's the beauty of this technology, and I think we're still working together to improve the process of breaking it down faster as well.
Should we quickly try and do a question?
So for me, because I haven't trialed it in I've only trialed it in glass houses, I only do 1 spray, and that lasts for 14 days in glass houses.
Thank you, Pratyush, and thank you, jury. Now, please welcome Mingji Yang of RMIT University.
Thank you. 1 in 20 Australian people are living with diabetes. Globally, diabetes impacts more than 400 million people. Once diagnosed with diabetes, the patients are required to track their blood glucose levels frequently to prevent complications, even death. But there are only 2 options for them, either by finger prick to withdraw the blood, or attaching a microneedle into the skin. Both methods are invasive, painful, and have risk of infection. Even worse, there is no cure for diabetes, making this painful management a lifelong process. Beyond causing pain, these two methods require consumables, either the test strips or the microneedle sensors. Both are generating biohazard waste. My solution is using light to detect glucose. The optical sensing has non-invasive nature, but what are the suitable wavelengths to do glucose detection? During my PhD study, I explored the spectroscopy of glucose in different solutions. I found 2 promising wavebands and pin out 4 peaks. Utilizing the largest 2 peaks, I developed a prototype, an optical glucose sensor. It is a miniaturized device with the dimensions just a few centimeters. The optical sensing technology can be further integrated into wearable electronics, such as a smartwatch, achieving continuous glucose monitoring. The optical sensing technology doesn't require any consumables, reduce both the cost and the waste. I hope in the near future, this optical glucose sensor can provide diabetes people with a more comfortable, easier, and cheaper way to detect glucose levels. Thank you.
Thank you, Minji. Over to questions, please.
Thank you, that was really good. This is probably one of the most competitive markets out there. It's a huge worldwide market. So who are your competitors in this market for this sort of technology? Because I'd imagine there must be many other people trying to devise similar sort of approaches.
Currently the dominator, I think, is the free library, that one, and the Dextom. And I can also share some information about the cost of these 2 methods. For the test strip, there are nearly 1,000 test strips per year for 1 patient. And that one costs around a few hundred. And the microneedle taps, they need 2 weeks to change a new one. So around 25 pieces per year. That one is nearly $3,000. And I think this one can, I hope this one can, like 1 year, just 1 device is fine because it's not consumed the optical sensing. And the cost will be much higher, much lower, sorry.
I have a second question.
I wanted to ask you, have you compared your method to the classical methods you had mentioned at the beginning? What is the precision of your method towards the other ones?
It is undergoing, yes. We are comparing our results to the other machines, and the clinical trials need some time. Yeah, but in the lab, we achieved this detect glucose very well. Yeah.
What?
We can't hear.
The comparable result is still undergoing. Yeah.
Can I ask, does it look through the skin? How do you actually see the glucose so that you don't have to get blood on the surface?
Yes, the optical sensing method is non-contact, so don't need to touch the molecule, so it penetrates into the skin. We promote several promising locations on the body. Yeah, that one, so the topic is breaking the wall of optical sensing, glucose sensing, but if, like, I find that I validated this a few promising locations on body, yeah, it's another big wall, I believe.
Okay.
Let's thank Minji. Good. And up next is Mehershad Wadia of the University of Queensland. Over to you.
the brain is the most important organ of our body. And you know it's true because it is my brain that's telling me that. Now, if you ever had a broken bone or a wound, you know that our body has a way to repair these tissues. And yet, for some reason, our body is unable to repair these important brain cells called neurons when they are damaged. You can think of neurons, bundled into nerves, as long electrical cables that connect your brain to the rest of your body. Now, a damage to one of these cables would lead to a loss of communication between the brain and its target, such as your limbs or organs. And our body's inability to repair these cables is directly linked to spinal cord injuries, paralysis, and other lifelong disabilities that affect more than 2 million people every year. Now, the human brain is complex and made up of billions of these cables, which makes treating individual nerve injuries very, very challenging. I know, if only there was some kind of magic glue that could get in there and join the two broken halves back together. And so, we looked at nature in its most basic forms to see how they solve this complex biological problem. Turns out, a tiny roundworm called C. elegans, present in your own backyards, has the remarkable ability of repairing its own nerves. And it does this in the most intuitive way that you and I can think of, by using a magical molecular glue called fusogens. Now, when neurons in these worms break, it spontaneously regenerates one of those broken halves till it reaches the other half. And then, by using this molecular glue, it's able to fuse the two halves back together, completely repairing the neuron and restoring its function. Isn't that amazing? Now, we have discovered the molecules that allows the worm to do this, and turns out that we humans actually have a version of this molecular glue that does not repair nerves yet. We also find that just like with worm neurons, even human neurons were able to fuse to each other as long as you provided them with this molecular glue. I'm currently studying these worm molecules and its repair mechanisms because that would allow us to re-engineer our own molecules and unlock their repair potential to treat some of our devastating nerve injuries. Being able to repair our own nerves would help break massive walls in human health and medicine. And a simple soil-dwelling roundworm might just be the key to transforming our permanent nerve injuries into temporary ones. Wouldn't that be magical? Thank you.
Thank you, Mehershad. Jury, any questions?
Very nice presentation, Mehershad. In terms of using these molecules, are there any side effects of unwanted things happening by injecting these ones?
There could be, because you could have neurons fusing to each other instead of repairing the neuron. But the mechanism that we saw, we identified molecules that act like signals. So when a neuron is cut, only that neuron says, hey, save me. And that's how the molecular glue knows where to go. So by using this mechanism in worms, where side effects don't happen, we can actually use the same mechanisms to make this very, very targeted, which would be very useful.
So what stage of research is in at the moment?
Right now, we've shown with human neurons on a dish. We obviously can't do a lot of these experiments directly in humans. So currently, we are doing this on a dish with human neurons. And then we would want to do this in mice, other higher mammals, and then clinical trials.
Okay, thank you.
About this, but I'm just curious. You said that we already have this particular chemical in our bodies already.
Yes, we have a human version of this worm protein, yes.
So do you have you got an idea of how you're going to be able to use that and make it work in a different way? What's your thoughts there?
Yes, so we know how the worm molecule looks, and we know how the human molecule looks. And we know the domains that make up the worm molecule, and we know how they're different from the human one. So by replicating the kind of domains that the worm molecule has, we can sort of re-engineer our own molecules to make it do those specific functions in a similar way.
Oh, that's really clever.
Any quick ones? We'll wrap it up there. Thank you, Mehershad. Now for our 15th presenter, Dr. Jonathan Ricchetti of the CSIRO.
Hi everyone. I'm here to break down the wall of the grain industry with hard data for a more sustainable production system. Australia is one of the world's top food suppliers. We are the biggest exporter of wheat in the globe, with more than $10 billion exported in 2022. And that's $3 billion more than the previous year. So we know we're growing more food, but are we doing it sustainably? While it's critical to continue to supply food for the world, we need to do it so in a way that we do not damage our environment and our nature. And this is what my research is about. How can we quantify our wheat production system is actually sustainable? Well, we measure it. We measure the gap between the maximal potential we could achieve and what we actually are achieving. And this is called the yield gap, which tell us how efficient our farmers are. The smaller the yield gap, the more efficient our farmers are. But that's a performance metric only. Now, if we marry that with a sustainability index, we can start to understand and metrify how our production system sustainably actually is. For example, if we take the water use efficiency, or how much food you can produce per amount of water, and link that with those metrics, we can start understanding the whole process. The less water we use, the more water efficient we are, the more water we save. So our team and I are combining lots of different data sources, from satellite imagery to weather data and soil information, to produce maps of how our production system is. And good news is that we are producing more food with less water. Our farmers are more efficient. With this important target, we now can think about the next steps. Other sustainability metrics of interest, like carbon, nitrogen, or ESG metrics, define the key drivers that are linked to those and determine the optimal sustainable practice for our grain industry. Thank you.
Thank you, Jonathan. Now, over to questions, please.
Thank you for this very interesting presentation. What are the costs involved with the method that you are using to find out these interesting facts?
Yes, so at this stage, we're working on the proof of concept. The main question we wanted to answer is, can we measure this at scale? So Australia is a continent, so can we do that to every farm in Australia and have that accuracy in numbers? So part of the next steps is to partner with companies that can do those kinds of pipeline to then evaluate how much this would cost. But we will need to do it because we need to measure if we're going to the right direction. So the quick answer, no, we have not looked at the costs yet.
What are you using that's different to be able to bring together these different data sources? Are you using new algorithms or AI or what, supercomputers? What's your process?
The answer is yes, all of them. So we have different satellite sources that are coming up. We have developed new algorithms, including AI models, but also traditional stochastic crop process-based modeling, and the algorithms to glue those informations together. So all of these parts are kind of developed.
Thank you very much. Are you matching from your satellite data with ground data evidence, such as moisture and from the farmers in the soil, etc.? So you're matching that data together with, to test it?
At this stage, because we are matching our, not only the satellite data, but the weather with, and soil with weather stations and some trials of farms, but mainly the ABS data is one of where we hold.
Great. Thank you very much.
Thank you.
And now for our final presenter. Please welcome Dr. Azara Senaratne of the Flinders University. Please.
Viruses, although they are tiny, they take a heavy toll on individuals, societies, and economies. So if we don't understand about these viruses properly, they can change the world, as we have already experienced. According to recent research published in the Medical Journal of Australia, long COVID alone costs Australia $10 billion in 1 year. So this is clear evidence that now more than ever, we must be studying about viruses from a broader perspective. Anomaly detection is one such technique that we can apply on viral genomics to study about the mutation and adaptation of viruses. While there are existing techniques that model virus life cycle, they all suffer from a significant drawback. That is, the dependence on a single observer or viewpoint for decision making. And as a solution, I'm working on developing a machine learning based system called the Co-Scientist, which tries to bring computer science at the service of health science. My Co-Scientist is a multidimensional anomalous space modeling technique that can define multiple observer points so that it can establish multiple normal and abnormal spaces in an evolving manner. An observer point can be any spectrum of variations, such as a set of rules or thresholds which you can use to define normality. And an instance that fails to obtain the membership of the normality of a particular observer point will be defined as an anomaly. So by studying all these normal and abnormal spaces, you can get an understanding of the progression of a virus in terms of its mutation, as it happens or even before it happens, and also the effectiveness of drugs in inducing cessation in a virus. So I work together with bioinformaticians than meets the eye. Thank you.
Thanks, Asara. Questions, please?
You're obviously putting them into a computer, the AI component of it. So what sort of grunt do you need for that computer? I mean, how big does it need to be, and the sort of cost we're looking at, and the time frame that it would take to analyze all these inputs?
So in terms of time-wise, now actually all the data comes from the virologists and the bioinformaticians, because the whole perspective is I, as a data scientist, I'm going to see how I can help them. And then, so they already have data that they work with, and with whether it's about millions of data that we talk about, or whether it's just few hundreds that they have, my aim is to see how I can adjust or bring in these different AI techniques to support them.
We can't.
Oh, yeah. Okay, so to be honest now, when it comes to me, it's all about whatever the analysis that the virologists or the bioinformaticians would like to study about. So they come to me with different observer points, I would say, and then I get their opinion and their insights in configuring these AI techniques.
Started talking about long COVID. Has your work actually shown any information that we can learn more about what that means and how we might be able to predict or be able to avoid people getting long COVID?
No, not really on that, because as I said, now, I would say the background of this, or why I'm motivated as a computer scientist to bring in this, is because I started, I would say, my PhD way back when the pandemic hit. And I was looking more at patient data to see patterns and the spread of COVID. But then I thought it's right time that I actually drill in to see how I can support the virologists to actually model the viruses. So again, a specific strain of a virus doesn't really matter to me. It's just that I'm here to support the virologists.
Thank you.
Thank you, Asara. Thank you, jury, for your diligent work. This actually now concludes the presentation and the Q&A. And isn't it a wonderful job that everybody did? Let's give them a round of applause to all of them. I personally feel very uplifted. How about you, Emma?
So good.
Yeah? But there is an important task to be done by the jury. And they will now head to the Becker room to deliberate and select the winner. Thank you, jury, and it's your work now. Everyone else, please make the way to the Jäger room, which is behind this wall out of the 2 days, for refreshments. Please also remember to vote for the People's Choice Award on your way out, and just vote once. We will all return to the theater at about 4:15 for the announcement of the winners. And please let me remind you, when you come back, food and drinks are not allowed in here. For those online, I hope you enjoyed all the talks as much as we did. And we will be back soon with you. In the meantime, enjoy fascinating science with upcoming video entertainment. Thank you.
So why does science matter?
Science is what drives our advances in medicine and what gives us new treatments, new vaccines, new techniques in surgery. All of those need to be evaluated, developed, many of which come from crazy ideas from fundamental science. Other advances come from astute observations by clinicians. So science is so critical to advances in medicine.
And I think this was one of the most beautiful examples of science in action, where every biomedical researcher on this planet over the last 3 or 4 or 5 years has read a paper and many have written papers about a virus that they had never heard of. I find this mind-blowing to what degree we have, as a community of scientists, come and tackled this new threat.
I'm motivated to research antibiotic pharmacology because I see the human cost of deadly bacterial infections. I think science is fundamental to discover the mechanisms, and then we can use the mechanistic findings to develop novel therapeutics. This kind of translation is not come out from a simple thinking. It come up with a long-term research. So that's why science is so critical.
Science is just the way that we learn to think. You know, we learn to gather data, interpret data, make decisions based on data. And over the last few years, it's been really surprising and horrifying, actually, to see that not everybody thinks that way.
We live in a world where we need to make informed decisions about how we build our houses so that they use less energy, so that they're more comfortable, healthier, better places to be. Even just down to the simple decision of, you know, whether or not you put insulation into your walls or into your roof, science can tell you the answer, which is the better of the two things to do.
You know, if you think about thousands of years into the future, without science, we don't have a great hope of survival, I don't think. You know, science will underpin all of that. A better future for our kids, you know, that's going to come from science.
Taking medicine in the future could be as simple as eating a salad.
The example that we're actually doing at the moment is to have a multiple sclerosis drug in tomatoes so that people with chronic multiple sclerosis could maybe have their Greek salad and have some level of medical protection from the disease.
These drugs are based on molecules known as peptides. They are smaller than proteins and carry out important biological functions such as regulating other molecules, and some even act as hormones. Several peptides have pharmaceutical potential, including as potent painkillers. But because most break down if they're taken by mouth, Professor David Craik and his colleagues have engineered them into more stable circular peptides called cyclotides. Cyclotides are naturally found in some plants, so Professor Craik's team can embed them into edible plants such as tomatoes.
One of the issues with current painkillers is that often they don't work, and then we also know with opioids there are horrible addiction problems in the so-called opioid crisis. This is a different class of molecule that doesn't have those addictive effects.
Professor Craik says the potential applications extend beyond chronic pain and disease to areas including agriculture.
We could be creating a new biofarming industry in Australia where farmers could be, rather than growing a crop that might be worth $100 a tonne, it could be worth $100,000 a tonne by producing a high-value medicine in those plants.
The innovation isn't far from becoming reality, with the first crops potentially commercialised in the next few years.
Obviously there's lots of regulatory and social and ethical issues that we would need to address in doing this, but I think it does open up a new world for combining agriculture and medicine and delivering medicines in a novel way.
For many animals, their sex is determined by their genetics. But researchers have found that the sex of some species at birth is determined by their environment.
They do it by temperature, and we've always wondered, how does that work?
Distinguished Professor Jenny Graves and collaborators at the University of Canberra have spent the last 2 decades studying this phenomenon in Australian dragon lizards. The lizard's sex is determined by sex chromosomes. When the eggs are incubated between 22 degrees Celsius and 32 degrees Celsius, the usual temperature range in the wild, ZZ eggs hatch as males and ZW eggs hatch as females. But when the temperatures rose above 34 degrees Celsius, the team noticed something strange.
Amazingly, what we found is at the higher temperature, the ZZ eggs, which should have turned into males, they turned into females. So they hatch as females, and amazingly, they're fertile.
Because these sex-reversed females can successfully mate with males, it has wiped out the female W chromosome.
So we've changed the whole sex-determining system. We've shifted it from genetic sex to environmental sex in one generation.
Temperature also drives sex determination in other reptiles such as alligators and sea turtles. It's a system that could have huge implications as the Earth's climate warms.
If global climate change keeps on getting hotter, we're going to have all-female lizards. We have all-female turtles and all-male alligators. Not going to work very well.
Scientists are now looking at whether different species could adapt to rising temperatures and avoid the disappearance of one sex.
The scientists say this probably means they are dealing with some new deadly sexually transmitted disease.
It's been 40 years since the first description of AIDS-related illnesses. We still don't have a cure or even a vaccine, but experts in the field say that doesn't tell the full story.
40 years seems a very long time. However, the progress in HIV has been extraordinary. I mean, in many ways, it's to me the biggest and most important scientific advance in the last century, because we developed a way of managing a virus that had a universal death sentence and make it a completely manageable chronic illness, all through science and the availability of antiviral drugs.
HIV is a virus that gradually destroys cells used by the body to fight off disease. It can severely damage your immune system, making it much easier to develop diseases such as tuberculosis and some cancers. The most advanced stage of HIV is known as AIDS.
Even in Australia, about 1 in 5 people diagnosed with HIV are diagnosed so late their immune function has deteriorated to a point where they have AIDS.
But overall, new cases of HIV in Australia have halved over the last decade. In fact, Sydney is on track to become the first place in the world to end community transmission of HIV by 2025.
It's time to make history.
Effective elimination is defined as a 90% reduction in new cases over a decade. The big improvement is largely due to high testing rates and preventative measures taken by gay and bisexual communities. But infection rates in the heterosexual population haven't fallen as sharply. The Kirby Institute's Professor Rebecca Guy says the data shows why regular sexual health checkups are critical.
The test is like the gateway to a whole range of things that are important for a person. So you think about HIV, if you have a test and you find out it's positive, you can start treatment straight away and you can live the same longevity of life of everyone else and remain healthy.
While accessing an HIV test is relatively simple in Australia, the same can't be said for many places in the world.
People at risk of HIV, men who have sex with men, for example, there are still countries that criminalise that activity. And so how can you get a public health message out if the activity is considered criminal?
Even where it's not illegal, the stigma around HIV can sometimes drive down testing rates. That's one reason the Kirby has led the push for more screening options, including self-testing kits for people to use at home.
They're not quite as accurate as a lab test, but I guess the way I always think about it is having a test, even if it's less accurate, is better than no test at all.
Professor Sharon Lewin is a Fellow of the Australian Academy of Science and President of the International AIDS Society. As head of the Doherty Institute, she played a key role in fighting COVID-19, a virus that we managed to have vaccines for within 12 months. So why haven't we been able to do the same with HIV?
Well, HIV is a very different virus to COVID or influenza, which are viruses that can be completely cleared from the body. HIV is unable to be cleared from the body, and that's because it's a virus, part of a family, that's able to become part of a person's DNA.
And that happens within hours of a person becoming infected. Treatments allow the immune system to recover and can even stop the virus from being passed on. But the virus doesn't disappear. It's just hiding and will re-emerge if treatment stops. Professor Lewin has dedicated her career to understanding this phenomenon, known as HIV latency.
I've been on the hunt for understanding where the virus hides, how to mimic latency in laboratory models, and ways to coax it out of hiding.
While we don't yet have a vaccine, we do have access to a medicine that can help prevent HIV, known as PrEP. If taken correctly, the daily tablet can lower the risk of infection by 99%.
In Australia, we have a public health system that makes medicine free, essentially. We have a fantastically mobilised, vocal and articulate gay community that have done a lot of the education around accessing PrEP, and our uptake of PrEP is one of the highest in the world.
PrEP doesn't offer protection from other sexually transmitted infections, so doctors recommend the use of condoms. Professor Lewin says PrEP is just one of many advances in HIV research that goes to show the power of science. She believes it's only a matter of time before HIV is cured once and for all.
It's become a very important, high priority area of research because we now have treatment, we now have widespread access to treatment. Treatment's relatively simple. The next big challenge is finding a cure.
The average Australian spends at least 90% of their time indoors. But how often do you think about the air you're breathing in? Indoor air pollution is considered a serious risk to public health in societies around the world. Despite that, most countries don't have any indoor air quality standards.
Every drop of water we drink from the tap, its quality is highly regulated. Every piece of food we take to our mouth is highly regulated in quality. Indoor air, which we take into our lungs 12 times a minute, is not regulated at all.
Professor Lydia Moravska from Queensland University of Technology was named one of Time magazine's 100 most influential people in 2021 for pressuring the World Health Organization into acknowledging that COVID-19 could be spread through the air, particularly indoors. Now she's leading a new push for international action. Professor Moravska and a group of global experts have used the prestigious journal Science to publish a blueprint for mandating indoor air quality in public buildings.
Our paper, which is a consensus of over 40 scientists and experts from many disciplines from around the world, is the best practice, better than anything else which is happening anywhere in the world.
The group proposes standards for 3 key indoor pollutants: carbon dioxide, carbon monoxide, and PM2.5, which are particles so small they can lodge deep in the lungs and enter the bloodstream.
This is my favorite little monitor. It measures carbon dioxide. Together with carbon dioxide, we emit other things, in particular pathogens, bacteria or viruses, if we are infected. So if concentration of carbon dioxide increases, potentially concentration of these pathogens increases as well. And it fits into my handbag, so it goes with me everywhere.
The scientists also want to ensure mechanical ventilation systems remove and dilute pollutants at a higher rate than their production. She points to schools as an obvious place for action, where many classrooms are now being fitted with air conditioning.
One issue with this is that while it provides a good thermal comfort, it does not provide fresh air. It just recirculates the air, so it doesn't do anything to improve air quality.
Outdoor air quality is already regulated in many places in line with the World Health Organization's global air guidelines. Professor Moravska says countries such as Australia can now lead by example for indoor air as well.
We know how to do it. We know how important it is. Why won't we start now?
Should a good bottle of wine be sealed with a cork or a cap? There was a time when the high-end wine world scoffed at the mere mention of screw caps.
It might seem a simple thing, but that required an enormous amount of research.
It was in fact industry leaders in Australia who led the push, eager to increase consumer choice. Research has shown that screw caps offer a better seal, meaning oxygen can't sneak into the bottle. The result is a crisper and more well-preserved wine, suited to most varieties of white. Some winemakers insist that fuller reds benefit from a little oxygen in the bottle and therefore age better with corks. But screw caps have another benefit. They prevent the wine from ever being corked, a term used to describe the presence of undesirable chemical compounds almost always transferred from poor cork batches. Corked wine has a characteristic odour that some compare to a wet dog.
We understand so much about the underlying science in Australia that we virtually get it right every time.
Professor Peter Hoy is Vice-Chancellor of the University of Adelaide, but he was also Australia's first Professor of Viticultural Science and then Enology. In other words, an expert in the science of grapes and wine.
This is where science always comes out on top. It enables things because people trust it.
At first glance, this could be a plantation of Christmas trees in the Australian outback. But you're looking at what some are calling one of humanity's biggest ever scientific endeavours. This is an artist's impression of what's known as the SKA Low Telescope, currently being built in Western Australia. It's one of two critical components that, when completed, will together form the SKA Observatory, or SKAO. The second part is the SKA Mid Telescope, now under construction in South Africa. SKAO will consist of more than 130,000 antennas and almost 200 dishes across the two continents. That will make it the largest radio observatory in the world, promising to answer some of our biggest questions about the universe. ANU's Professor Naomi McClure-Griffiths is a Fellow of the Australian Academy of Science who discovered a spiral arm of the Milky Way and is chair of SKAO's Science and Engineering Advisory Committee. She says SKAO will enable science to take a giant leap forward.
It's the chance to be able to see the very first stars in the universe and when those turned on. And then everything from the very beginning to us here and looking at how planets form in our own galaxy.
For that to happen, radio telescopes must be located a long way from other human-made transmissions, such as TV, radio, and mobile phone signals, which can interfere with the relatively weak radio waves coming from space. That makes this site on Wajiri country ideal. It's already home to precursor telescopes like this one. The site itself is called Inyarimana Ilgari Bundara, the CSIRO Murchison Radio Astronomy Observatory, which translates to sharing sky and stars. The traditional owners agreed to host the science facilities on their land as part of a broader agreement that will ensure educational, social, and economic benefits for community. And the benefits to society as a whole can't be overstated. Science Minister Ed Husick says SKAO's cutting-edge technology will expose Australian businesses to new skills and capabilities. We will see those changes flow on for generations to come. SKAO will also enhance Australia's growing space industry, with scientists from around the world making regular use of the observatory. Some will even be on the lookout for intelligent extraterrestrial life. If an alien species were out there trying to reach out and say hello, by the time that radio signal got to Earth, it would have been so weak we wouldn't be able to detect it, says SKA Low Telescope Director Dr. Sarah Pearce, but that changes with SKAO. The observatory will start producing science before the end of the decade. You're watching time-lapse footage of dung beetles at work. Their remarkable ability to remove dung makes them the perfect biocontrol agents. Research has shown their presence significantly reduces the number of flies and other pests, while also improving pasture growth and soil retention. So, scientists like Professor Leslie Weston have been searching the world for the best species to bring home. But wait, doesn't Australia already have dung beetles?
We have 500 native dung beetles, but those dung beetles are geared to sourcing marsupial dung or drier, small, hard pelleted dung, whereas when cattle, sheep, horses and other livestock were introduced to Australia, they generate a larger, wet, moist paddy.
CSIRO began importing dung beetles to Australia in the 1960s. The program has been overwhelmingly successful. However, the introduced species haven't adapted to all regions of Australia, and some will simply stop working during certain periods of the year. Researchers are working to address these gaps with a joint venture called the Dung Beetles Ecosystems Engineers Project, supported by Meat and Livestock Australia, with federal funding from the Department of Agriculture, Fisheries and Forestry.
So we'd like to have a full complement of dung beetles that are active all year round, because obviously cattle or sheep are producing dung all year round that needs to be.
Their strict quarantine regime is over. The offspring will then be distributed to field sites across southern Australia.
This farm near the New South Wales city of Wagga Wagga is a long way from war-torn Ukraine.
These varieties here, they're all spring canola, and in Australia we mostly grow spring canola.
But it's familiar territory for Marina Tsu and Svetlana Omulchuk.
Big surprise for me that we have in Ukraine and in Australia. It's really was like, Australia was something very fine, different from us, and when we are here, we look and wow, we have the same thing. So we are very interested in.
Marina and Svetlana research canola at the Ukrainian Scientific Institute of Plant Breeding in Kyiv.
For both countries, it's an important export crop, and for the globe, countries like Australia and Ukraine that basically provide the world trade of that commodity, what happens in those countries, in both of our countries, is important to world trade.
And it's what's happening in Ukraine right now that has brought these young scientists to Australia. When war broke out, the US-based Breakthrough Prize Foundation teamed up with the Australian Academy of Science to ensure Ukrainian researchers could continue their work.
Ukraine is actually a powerhouse, particularly in genetics and in plant science, so it's important for us to keep those links open. But whatever helps Ukraine also helps Australian science and international science.
Like everything, the work of scientists has been seriously interrupted by the conflict.
It's impacted all spheres of our life, not only science, because you always are under stress, you're always worried about safety of yourself, of your family, your friends, and it's hard to plan your experiments and projects when you think just how to survive.
Життя змінилося, але ми якось продовжуємо працювати. Дуже вдячні всім, хто нас підтримує, всім країнам, також Австралії, які надають допомогу.
One of their hosts in Australia was CSIRO agricultural scientist and fellow of the Australian Academy of Science, Dr. John Kierkegaard.
They told me sometimes they're travelling along roads which have mines. It takes them hours, 10 or more hours to travel safely to see some of their field experiments. Quite challenging, and I found them remarkable, but in their pragmatism and determination to sort of keep the work going.
Dr. Kierkegaard had hoped other Ukrainian scientists would join the Australian trip, but fighting-aged men have been barred from leaving Ukraine. Australia has made sure they still have support, including remote access to scientific infrastructure, including supercomputers, microscopes and telescopes.
Science and technology has clearly underpinned the growth of canola as a crop in both countries. The only way to sort of stay ahead of that game is that scientists around the world collaborate, share ideas, share information, and that's what conferences like this and exchanges like this can really promote.
Ukraine knows science will play a major role in rebuilding the country after the war.
And it's all about the human capital. We can rebuild buildings, right? It's not a problem, but we need to keep our human capital. It's our competitive advantage, the talent that we have. And we need to nurture the talent. We need to help the talent to grow, develop, and become better. And I think this will be of huge contribution to Ukraine's reconstruction eventually.
The support of Australia and Australian people are really important for us and for Ukraine now, because I know it's important to feel that we are not alone in this need. And we really appreciate all that you do for us.
You're looking at the largest single dish radio telescope in the world. It's called FAST and located within the mountains of southwestern China.
And that is by far the most sensitive radio telescope in the world. I first went there while they were putting the first few things on the ground, and it was amazing to see how quickly in China you'd come back a year later and the whole dish would be built.
At the heart of FAST is a primary multi-beam receiver designed and built by Australian scientists. China chose to collaborate with Australia because of our experience with the Parkes radio telescope in regional New South Wales.
The Parkes radio telescope is by far one of the greatest telescopes in the world. It's been
who help make this event possible. And to start with, please welcome Josie Downey, General Manager and Managing Director of Merck Healthcare Australia, New Zealand, to address all of us. Josie.
Thank you. Honourable members of the Academy, Her Excellency, distinguished guests and talented young scientists, I'm absolutely delighted to be here today, and I'm inspired by the life-changing innovations that have been presented today. I'd like to acknowledge the traditional custodians of the land on which we're meeting today, the Ngunnawal people and recognise any other people with connection to the lands in this region. I respect their continuing culture and the contribution that they make to the life of this city and this region. As a STEM champion, I can't think of a better way to spend a Friday. At Merck, science is at the heart of everything we do. In healthcare, we create, improve, and prolong life. We focus on challenging diseases such as cancer and multiple sclerosis, and through our fertility expertise, we enable people to become parents. Through life science, we enable the scientific community. And through electronics, we are the company behind the companies advancing digital living. We have a presence in 66 countries around the world, and I'm very proud to say that we have a presence in Australia and New Zealand. And as a German science and technology company dedicated to health, we are especially proud to be supporting Falling Walls. Over the millennia, science and technology have advanced remarkably, but the key challenges remain. And this underscores the value of today's event. And observing the best and brightest scientists, accomplished innovators, present their work is absolutely inspiring. And it presents to us the potential for a better tomorrow. And now more than ever, we need critical thinking and evidence-based science in order to enable the dreams of tomorrow. I'm truly inspired by the ideas that were presented today, and congratulations to those scientists on your achievements and for getting to this event today. Best of luck to all of you.
Thank you, Josie, and thank you for these thoughts and also for the support you're giving for the scientists. And one of the things we can do with this sponsorship is send the 3 winners to Berlin, which is the important part that will happen in November. Now, we also have a sponsor who represents more or less Europe, in the sense of EURAccess is a company, an organization, I should say, that is looking after the links between Europe and, in this case, Australia in research. So we have a video message from EURAccess.
Good afternoon, everyone. I hope you are having a good time listening to the amazing finalists of the Falling Walls Australia competition. Unfortunately, I could not be there in person, but in essence, I'm there listening to all of your pitches and wishing you all the very best for the competition. I'm Nishan Sandilia. I'm the Regional Coordinator for Australia and New Zealand at EURAccess. EURAccess is an initiative of the European Commission that addresses barriers to the mobility of researchers and enhances scientific collaboration between Europe and the rest of the world. We try to help researchers who are interested to learn more about European funding, career opportunities, international collaboration, and bridge the gap that there exists between Europe and Australia. Now, with Falling Walls, we decided on a long-term partnership with the Australian Academy of Science, and it's our 5th year of collaborating with the Academy for the finale event in Canberra. We are also organizers of the Falling Walls Sydney event that took place earlier in July. It's excellent to be part of this initiative, and as a gesture of your hard work, of the finalists' hard work, we'll invite the winners of the competition to a science communication and training program. The intention of this program will be to help the winners of the Australia contest in Berlin to fine-tune the nitty-gritties of how you pitch, how you compete in a competition like this. We will also facilitate connections desired by the winner in Europe should they want to establish contacts. This is possible because we have around 600 plus EURAccess centers in Europe, and we have access to a large number of European Research Council grantees, as well as Marie Skudowska-Curie fellows. With that, I'll not keep you waiting because I'm aware that you are all gearing up to hear who the winner of the contest is. I wish you all of the best, and I hope you have a good time ahead in the networking session as well. All the very best. Thank you very much for listening.
So as you heard, EURAccess has been a strong supporter for many years, and also for several years when the competition in Sydney. And it's important now that we have a long of support that you go out and tell people how good this is, because we like to maintain this level of the 5 state-based events, followed by the finale, and then, most importantly, by 3 people going to Berlin. Now, that gets me to the question of who are the audiences? Who do we actually want to address? Here we have a national event, and you are a great audience, both here in the room and online. And you might be thinking about what's good for Australia, for society, for all of us. In Berlin, we have a very diverse audience, and we have a very diverse jury. Emma, any thoughts about the audience that you actually encountered in Berlin?
Absolutely. I think that the magic of Falling Walls is your ability to broach past your area of specialty. I am from a medical background and only ever attended medical conferences where you can sometimes be in an echo chamber of people who have similar experiences or ideas to you. And I think that Falling Walls is just such an incredible opportunity to meet people from engineering, from chemistry, from physics, and get their ideas and also contributions to my work. And I think that's just such an interesting way to approach science, in being true collaborators, and often existing in silos only works against our science. So it was an incredible opportunity to speak to a broader audience, to progress my work, but also to contribute to others. And I think that was just such an amazing part of it. Another incredible part was discussing with industry leaders and just getting valuable input into how we can translate these ideas into the clinic, into the community, to help break down some of those walls.
Now, in the break, someone asked me, you were on the podium, right? Like you were one of the medal winners last year. Did you get a check or did you get a prize? Or what was the prize that you actually received?
There was a check, but more than that, it was an incredible opportunity to go to such an incredible country which really champions science, and the opportunities for postdoctoral work and industry and collaboration. That was truly very meaningful. And thank you to our judges for coming back.
Yes. And applause to our judges. They did the hard work. You want to invite Cathy to sit? Yeah. Good. Now, let me please invite Cathy.
Yes, okay.
And you're already here, right? Also...
Only because it's so hard to get out of those chairs.
Yeah. Also, Her Excellency...
I think she's on her way back.
She's on her way back. And Professor Jagadish to the stage. And what we will do is present participation certificates.
Oh, okay.
So we are not... Please stay. Mm-hmm. So we are not yet revealing.
Okay.
Good. Now, the best position might be with the posters.
Indeed.
Mm-hmm. And we're waiting for Excellency. And could you help me?
Absolutely.
When I read out a name, hand it on to them. So when we are ready, this looks a little bit rare. So I hope we get through that very quickly. Right? So unfortunately, yes. So we would like to present the certificates of participation to all of these presenters. So please, when you hear your name, make your way to the left of the stage and collect the participation certificate. And importantly, pause for a photo. Good. So we start with Dr. Stephanie Allison-Logan. Great, thank you. And followed by Isaac Kyei Barffour. And now we have Nicola Barishev. Thank you. Followed by Benjamin Kamer-Pesky. And while we're taking the photographs, this gives you a great opportunity to remember all these great ideas. And there's many of them. Now, Dr. Natasha De Alves. And we have Dr. Jacob Martin. And we have Dr. Donovan Garcia-Cerron. Followed by Kira Pignell. Saisam Ramesh, please come on stage. And Dr. Harshal Patel. And let's remember, all of them have already won one of 3 places in each state. The tension is building. Please, Dr. Kelly Matirovich. And we have Pradyush Ravishander. Followed by Minji Yang. And we have Mehershad Wadir. And Dr. Jonathan Riccati. And Dr. Asara Sanaratne. Good. Thank you so much, and a round of applause to all of you. Thank you, jury, for doing a great job. May I ask Dr. Cathy Foley to announce the winners? I think use this microphone if you like.
Yes, that'd be great.
And here's the paperwork.
All right, fantastic. All right, so can I just, oh, I see, okay.
The People's Award.
Yeah, no, I'm just getting that. All right, so first of all, this is really hard. It was just such a delight to hear everyone's stories and their presentations. I think Australia is in good shape with the extraordinary quality and innovation and willingness to participate too. All these things come together into something which is a very special afternoon. In fact, it's amazing how much information you can impart in 3 minutes with 2 minutes of questions. I think all of us have something to learn from that, possibly. Anyway, so let's go to start with the Audience Award. and it's, I'm going to really mangle people's names, so I'm really sorry if I get this wrong, but the audience award goes to Nicholas Barishev. The next one, we bring it over here.
Can we get this going? Yes, you're on here.
So third place goes to Pratish Ravichandra. I'm just going to, that's D. I'm just trying to decipher the writing. Okay, and the second place goes to Natasha de Cloyce, is it? Is that right? First place goes to Jacob Martin.
Well done, everyone.
Hey.
Congratulations to all of our presenters, and in particular, our winners. I wish you all the best for the Falling Walls Global Finale in Berlin in November. Please know that we'll be watching on from Australia and cheering you on. Enjoy every moment.
On behalf of the Academy, I would like to thank all our presenters for putting yourself forward for this opportunity. It's not that easy, but they did a terrific job. To our jury members, thank you for giving your time to judge today's presentation. What a great outcome. I would like to thank our event partners and sponsors once more for making Falling Walls Lab possible, the Merck Group, the Embassy of the Federal Republic of Germany in Canberra, and Euroaccess Worldwide, Australia, New Zealand. Many thanks to you, Emma, for joining me on stage today. It has been great fun. And finally, thank you for being part of the audience and for listening to these wonderful presentations. To those online, will we say now goodbye? And to those in the theater, please join us for drinks and canapes in the Jaeger room again. We will also be joined by members of the Australian-German Research Network who are kindly hosting and sponsoring the reception. But before you leave the theater, could I kindly ask you, everybody, to assemble here on stage and in front of the stage. We're sending our photographer up on there, and we like to have a big group photograph and say, hello to Berlin, we are coming. Please come up. We fill the stage, please.
So many amazing
Cathy, you want to come up? It doesn't really matter in which order, but you know.
Yeah, I was about to say, I should show you should be in the front.
Get at the back with the tall people.
Yeah, that's right. And come forward to come to the friends.
I think so,
Lock
yes.
everyone out.
Try to do that.
Yeah, it's a good idea.
Yeah.
Good idea.
Great to see you back this year.
Oh my gosh, they can't get rid of me. I'm hanging around like that. I'll be back next year.
No, it's so happy. It was so happy.
Oh, I've loved it. It's been so incredible. I'm just so excited.
So all of you also come, please.
No excuses.
Everybody.
How was your last visit? Well done.
Good.
I feel like I'm now seeing you then.
We're to Melbourne. Oh, yeah. 6:30.
Oh, yeah.
I'm very cool. We need to fill the gaps, I think.
Yeah,
They're
come on,
asking
fill in
us
the
to
gaps.
get in.
No.
Done it before.
Here he is.
Stop waving.
Yes. No pressure turns in.
How are you looking?
There's one up there as well. They're all alike.
Wave.
All right, we're waiting to Berlin. We need to be speaking German.
Yeah.
Royal way.
Yeah. That's what we practice.
Okay, thank you. Thanks, Cathy. Join you. Ambassador, thank you very much. Thank you. It's wonderful. It's wonderful. Yes. Pleasure meeting you.
You have to get into